Good morning friends. Today I will be talking to you about stem cells. Stem cells are progenitor immature precursor cells which are present all over our body in all our tissues, and these play a role in giving rise to all types of cells in our body. These are very important types of cells because from these cells all tissues and organs of our body arise. Right from the stage when the embryo is developing up to the adult stage, these stem cells play a very important role in regenerating and repairing our tissues. The stem cells basically are of three types based on their capacity to give rise to either the whole organism or to different types of cells and organs. The three types of stem cells are the totipotent stem cells, which can give rise to the whole organism as such, that is to the whole embryo as such, and then the pluripotent stem cells, which can give rise to practically all types of cells in our body. The multipotent stem cells, which is the third type of stem cells, can give rise to a limited range of cell types in our body. Stem cells are also divided into another three types based on the source from which they are taken. These are the embryonic stem cells and the adult stem cells. There is a third type of stem cells which is called as the embryonic germ cells, but they are of not much use today as it is very difficult to obtain them. The embryonic stem cells are pluripotent stem cells, that is they can give rise to all types of cells in our body, and they are basically obtained from a 5 to 7 day old embryo which is known as a blastocyst. The adult stem cells, which are also known as somatic stem cells, are obtained from any part of the adult human body, such as the brain, skin, liver, pancreas, and so on. I will be talking more in detail about the embryonic stem cells. Before that, let me give you a short summary of the adult stem cells. The adult stem cells are further divided into three types: the hematopoietic stem cells (HSCs), the mesenchymal stem cells (MSCs), and the somatic stem cells (SSCs). The hematopoietic stem cells are obtained from either the peripheral blood or from the bone marrow. These cells are capable of giving rise to all types of blood cells in our body. The mesenchymal stem cells are derived from the bone marrow. Either it can be the adult bone marrow or even the fetal bone marrow. These cells are capable of giving rise to many different types of cells in our body. The somatic stem cells are obtained from all other organs except the blood and bone marrow in the adult human body. There is a very special character for the adult stem cells. Apart from showing differentiation, they also show transdifferentiation, which is a brain stem cell can differentiate into different types of neurons. But instead of that, they can transdifferentiate into muscle cells or skin cells or liver cells. This would be of great help to us when it comes to stem cell therapy. Now friends, I will tell you more in detail about embryonic stem cells. Embryonic stem cells, as I've told before, are obtained from the inner cell mass of the human blastocyst. These inner cell mass, as shown in the picture, are a group of cells from which all body cells arise as the embryo develops and the baby is born. Embryonic stem cells need very special culture conditions to grow in the lab. The main culture condition for embryonic stem cells is that they need 95% oxygen, 5% carbon dioxide, and they have to be given a temperature of 37 degrees centigrade. This culture condition can be obtained in the lab by the use of the carbon dioxide incubator. Furthermore, the medium that is used for the culture of embryonic stem cells is a combination of the DM-EM medium, Dulbecko's modified Eagles medium, and 5 to 10% fetal bovine serum or fetal calf serum. DM-EM medium is available commercially in a very purified and sterilized form. Same way, the fetal calf serum or the fetal bovine serum is also available in a sterile form commercially. These can be ordered and obtained, used in the lab under sterile conditions. Along with the DMEM medium and the fetal bovine serum, we should also add glutamine, the amino acid, which is very essential for the growth of embryonic stem cells. Plus, we should add the antibiotics such as streptomycin. This is to prevent any microbial contamination during the growth of the embryonic stem cells in the lab. The feeder cells are very important for the growth of embryonic stem cells. Embryonic stem cells are not grown directly on a culture dish. Mouse embryonic fibroblast is grown on the dish as a base upon which the embryonic stem cells are grown. The feeder layers are usually fibroblast cells, which are also precursor cells, which is present throughout the body and which give rise to the cartilage and bone cells as well as the skin cells. Mouse embryonic stem cells were the first type of embryonic stem cells to be cultured. This was done by Martin Evans and Matthew Kaufman of the University of Cambridge. And these people were the first ones to show that mouse embryos could be cultured in the lab successfully, and then the inner cell mass could be isolated from the blastocysts of these mouse embryos. Gail R. Martin is another very important person in this field of embryonic stem culture because she was the first one to coin the term embryonic stem cells. She also proved that embryos can be very well maintained in the lab, and now we have very standard protocols for the maintenance of embryos as well as embryonic stem cells in the lab. Human embryonic stem cells were first cultured by James Thomson, and he also was the first person to successfully culture human embryos in the lab. Before him, nobody had ever attempted to culture and maintain human embryos in the lab. I have given the pictures of the morphology of mouse embryonic stem cells and human embryonic stem cells. In this comparison, you can see that the mouse embryonic stem cells have a rather irregular colony morphology. The outer boundaries of the colonies are very irregular compared to the human embryonic stem cell colonies, which are more rounded and regular. As we grow embryonic stem cells in culture in the lab, it is very important for us to note and to observe if they are undergoing differentiation. It is of utmost importance that we maintain our embryonic stem cells in the embryonic stage itself, that is as pluripotent stem cells. In case they show differentiation, then they are tending to become more mature and we can no longer call them embryonic stem cells. Now, there are many ethical issues related to the use of embryos to obtain the embryonic stem cells. Many of the pro-life activists are against the use of the embryos to collect stem cells, although these stem cells are very useful in the therapy of many disorders. We have noticed globally worldwide protests by the pro-life activists for and against the use of these embryos in stem cell therapy. So based on these protests against embryonic stem cells, many researchers took up research as to whether embryos could be avoided to obtain stem cells. For example, we can use the adult stem cells in stem cell therapy instead of using embryonic stem cells. The only disadvantage would be that embryonic stem cells, being pluripotent, would give rise to all types of cells in the body, so they could be used for all types of therapy and disorders. Whereas the adult stem cells are very limited in their capacity. As I've told before, they are multipotent and can give rise to only a specific number of cell types. So they probably cannot be used for all sorts of therapies. Therefore, in 2009, the researcher from Japan called as Dr. Shinya Yamanaka has arrived and created induced pluripotent stem cells. Induced pluripotent stem cells were created by Shinya Yamanaka by taking the adult skin fibroblast cells which have already matured and are already fully differentiated and introducing the developmental protein genes into them. Usually in our adult cells which are fully mature and differentiated, the developmental protein genes are all shut down because they are no longer needed for the cells to develop or to give rise to other cell types since they are already fully mature. But what Yamanaka did was, he took the skin fibroblasts from adult cells and then he introduced the genes for the developmental proteins which reprogrammed these cells, these adult mature cells, into becoming embryonic pluripotent cells. So these adult cells actually started behaving like embryonic cells. These cells are called as the induced pluripotent stem cells. Induced pluripotent stem cells created by Yamanaka would be a good source of pluripotent stem cells for stem cell therapy. In this issue, the main advantage of using induced pluripotent stem cells is that we are avoiding the use of embryos. So long, the world thought that pluripotent stem cells could only be obtained from embryos. But with Shinya Yamanaka's discovery of the induced pluripotent stem cells, we can now obtain pluripotent stem cells for stem cell therapy even from adult cells. And this is of great use and now being tried out in many clinical trials for the treatment of many disorders. Dear friends, it is very important for us to note that stem cells are of great importance in treating and giving life to many patients with many different disorders. A few examples of this is the treatment of cancers and immunodeficiency disorders. The stem cell therapy is also known as regenerative medicine because in patients who have lost their cells or organs or who have damaged cells or organs due to many different disorders can regain back their lost organs or cells by taking a transfusion of stem cells which have been grown and maintained in the lab. This is known as regenerative medicine and it is giving life to many people throughout the world. For many disorders, stem cell therapy or regenerative medicine has been tried out successfully. One example is the treatment of Parkinson's disorders in mice, which has been completely reverted by the administration of neuronal cells which were obtained from pluripotent stem cells in the lab. Diabetes, for example, where the pancreatic beta cells fail to function, have been cured by taking stem cells and differentiating them into pancreatic beta cells in the lab. Stem cell therapy can be done by taking stem cells in the lab, culturing them, storing them for long periods. Whenever needed by a patient, the stem cells can be taken, thawed and then differentiated in the lab into many different cell types of our choice by giving the appropriate differentiation factors. Whenever we want a heart muscle cell or a cardiac muscle cell, we can make a pluripotent stem cell to differentiate into a cardiac muscle cell. This cardiac muscle cell can be used for a patient who has suffered heart damage. In the same way, a pluripotent stem cell which is cultured in the lab can be made specifically to differentiate into neurons and given to Alzheimer's patients who have lost their neurons in the brain. Dear friends, there are many more examples of stem cell therapy which I can say. For example, even the treatment of baldness, in which stem cells are implanted in the hair follicles so that they give rise to new hair. They are also used for anti-aging treatment where old persons can renew their cells within the body which have aged out. Dear friends, I hope this has been very useful to you in gaining a basic knowledge of stem cells and the way they are used in stem cell therapy. Thank you.